The organization and expression of Ig genes. The vertebrate immune system is capable of responding to an essentially infinite array of foreign antigens. Variable versus constant regions are their basis for variability, organization, and expression of Ig genes. Historical perspective, early theories, multi-gene organization of Ig genes, variable region rearrangements, basics of mechanism like key players, DNA signal sequences, and specific enzymes. Then generation of AB diversity, 7 primary roots. Bone marrow, peripheral lymphoid organs, cell Ig expressed. Historical perspective, Ig sequence analysis revealed many dilemmas. Extreme diversity of AB specificity, variable regions versus constant regions, isotype with similar Ag specificity but differing, heavy chain constant regions. Then historical perspective. Proponents of the one gene, one protein paradigm had trouble reconciling this model with the oddities of IGs. This led to an initial germline theory which suggested that a significant portion of the genome is dedicated solely to AB code. This led to an initial germline theory which suggested that a significant portion of the genome is dedicated solely to AB coding. The immune system is that important. Then historical perspective. In contrast, somatic variation theories emerged which suggested the opposite. Relatively small amount of Ig gene specificity arises from mutation or recombination. Dreyer and Bennett in 1965, 2 gene, 1 protein model. No precedent in any biological system. Then Tonegawa and Koshumi compare IgDNA from embryonic, germline, and adult myeloma somatic cells. Compare IgDNA from embryonic, germline, and adult myeloma somatic cells. Experimental data suggested that during differentiation, the V and C genes undergo rearrangement. Multi-gene organization of Ig genes. Each class of Ig components, kappa, lambda, heavy, encoded by separate multi-gene families on different chromosomes. Each family contains several coding sequences. Each class of Ig components, kappa, lambda, heavy, encoded by separate. Each class of Ig component, kappa, lambda, heavy, encoded by separate multi-gene families on different chromosomes. Each family contains several coding sequences or gene segments. Multi-gene organization, lambda, light chain, human 22, mouse 16. K light chain, human 2, mouse 6. Heavy chain 14, 12. Don't get confused. Lambda kappa 12, 2. Mouse lambda and lambda 16, kappa 6. Heavy chain in case of human 14, in case of mouse 12. Multi-gene organization of Ig genes. Kappa and lambda light chains, V variable, J joining, and C constant gene segments. Heavy chains, V, D diversity, J and C gene segments. A leader sequence also precedes each V segment. Kappa and lambda light chains, V variable, J joining, and C constant gene segments. Heavy chains, V, D diversity, J and C gene segments. A leader L sequence also precedes each V segment. Gene segments discovered by comparing DNA sequences with amino acid sequence of IGS. Organization of Ig germ-like gene segments, mouse. Variable region rearrangement, multifaceted process produces mature B cells which are committed to express specific AB. Specificity of AB determined by the sequence of its rearranged variable genes. Specificity of AB determined by the sequence of its rearranged variable genes. Light chain rearrangements, VJ rearrangements. Specific allowed rearrangements differ from species to species, but a big picture view can suffice. Rearrangement occurs in ordered steps but can be considered as random events which result in the random determination of AB specificity. Heavy chain rearrangements requires two separate rearrangement events like VDJ joining and VDJ joining. Differential polyadenylation and RNA splicing can result in mRNA with either CU or C delta. Heavy chain genes. B cells can express both IgM and IgD with identical AG specificity on its surface. Recombination signal sequences direct recombination. Mechanism of variable region DNA rearrangements. On one turn, RSS can only join with the two turn RSS. Why might this be? Gene segments are joined by a class of enzymes called recombinase. Two recombination activating genes encode proteins which act together to mediate VDJ joining. RAG1, RAG2. Mechanism of variable region DNA rearrangements. Rearrangements may be productive or non-productive. Allelic exclusion. Heavy chain genes only expressed from one chromosome. Light chain genes only expressed from one chromosome. Essential or for non-specificity. Expression of both alleles would result in a multi-specific P cell. Allelic exclusion. Generation of AB diversity. Multiple germline gene segments, combinatorial VDJ joining, junctional flexibility, P region nucleotide addition, N region nucleotide addition, somatic hypermutation, combinatorial association of light and heavy chains. Junctional flexibility, PN by N addition, somatic hypermutation, nucleotide replacement mediated by activation induced nucleotide replacement mediated by activation induced cysteine deaminase AIFD also plays a key role in class switching. Frequency of 10 raised to -3 per BP per generation. Somatic hypermutation. Next is about antibody engineering. A number of discoveries and technologies have converged to produce a renaissance in antibody therapeutics. Our vision of antibodies as tools for research useful for the prevention, detection, and treatment of disease has been revolutionized by these recent advances. This review specifically focuses on what is now called antibody engineering and includes chimeric and humanized antibodies, immunoglobulin fragments, antibody libraries, antibody fusion proteins, and transgenic organisms as bioreactors. As a consequence of refinements in antibody technology, the field of genetically engineered immunoglobulins has matured into an elegant and important drug and reagent development platform. Antibody proteins that bind to a specific target molecule, like antigen, at one specific antigenic site. In response to either infection or immunization with a foreign agent, the immune system generates many different antibodies that bind to the foreign molecules. Individual antibodies within this polyclonal antibody pool bind to specific sites on a target molecule known as epitopes. Epitope is on antigen. Okay. So isolation of an individual antibody within the polyclonal antibody pool would allow biochemical and biological characterization of a highly specific molecular entity targeting only a single epitope. Realization of a therapeutic potential of such specificity launched research into the development of methods to isolate and continuously generate a supply of a single lineage of antibody, a monoclonal antibody. In 1974, Kohlher and C. Milstein developed a process for the generation of monoclonal antibodies. In their process, fusion of an individual B cell or B lymphocyte, which produces an antibody with a single specificity but has a finite lifespan, with a myeloma B cell tumor cell, which can be grown indefinitely in culture, results in a hybridoma cell. This hybridoma retains desirable characteristics of both the parental cells, producing an antibody of a single specificity that can grow in culture indefinitely. So, generation of monoclonal antibodies through the hybridoma process worked well with B cells from rodents but not with B cells from humans. Consequently, the majority of the cells first monoclonal antibodies were from mice. When administered into humans as therapeutic agents in experimental tests, the human immune system recognized the mouse monoclonal antibodies as foreign antigens, causing an immune response which was sometimes severe. Although encouraging improvements in disease were sometimes seen, this response made murine mouse antibodies unacceptable for use in humans with a functional immune system. Fueled by advances in molecular biology and genetic engineering in the late 1980s, efforts to engineer new generations of monoclonal antibodies with reduced human immunogenicity have come to fruition. Today, there are a number of clonal antibodies approved for human therapeutic use in the United States. So, characterization of the structure of antibodies and their genes laid the foundation of antibody engineering. In most mammals, each antibody is composed of two different polypeptides, the immunoglobulin heavy chain IgH and the immunoglobulin light chain IgL. Comparison of the protein sequence of either heavy or light antibody chain reveals a portion that typically varies from one antibody to the next, the variable region, and a portion that is conserved, the constant region. A heavy and a light chain are folded together in an antibody to align their respective variable and constant regions. So, the unique shape of the co-folded heavy and light chain variable domains creates the variable domain of the antibody, which fits around the shape of a target epitope and confers the binding specificity of the antibody. Mice genetically engineered to produce fully human antibodies allow the use of established hybridoma technology to generate fully human antibodies directly. Mice generally engineered to produce fully human antibodies allow the use of established hybridoma technology to generate fully human antibodies.